Okay, so in this video, we are going to discuss Chapter 6 on photosynthesis. We are going to look at Part E on multiple choice question. For question number 1, the reaction center in photosystem 1 is also known as P700. At the reaction center of photosystem 1 or PS1, it has a special pair, of chlorophyll A, that will absorb only red light in the range of 700 nanometer. Question number 2. The following is or are the products of cyclic photophosphorylation. Cyclic photophosphorylation will only produce ATP through the process of the flow of electron and also chemiosmosis. So the electron will flow from the reaction center, the reaction center of photosystem 1, which is P700, and then it will pass from one electron carrier to another electron carrier and then back to P700 again. So the electron will be passed from the primary electron acceptor here and then through paradoxine and then to cytochrome complex and then to plastocyanine and then back to P700. And the passing of electron, the electron will be transferred from one carrier to another carrier, and the electron will release energy. So this energy will be used during chemiosmosis, to produce ATP molecule. The electron will release energy so that the energy can be used by the process of chemiosmosis to produce ATP molecule. Question 3. Pineapple is known as chem plant because it fixes carbon dioxide into organic acid at night. So for chem plant, the stomata will open during nighttime. The stomata will open during nighttime so that the carbon dioxide can enter into the leaves and can be fixed during the nighttime. So for chem plant, it fixes carbon dioxide twice. So the first fixation of carbon dioxide will occur at night, which involve the carbon dioxide to be incorporated with carbon dioxide acceptor, which is PEP, phosphoenol pyruvate, and then it will be converted into oxaloacetate. So this all happens at night, and then from malate it will be converted into from oxaloacetate it will be converted to malate. Malate that will be stored in the vacuole. Stored in the vacuole as malic acid. So that's why here it says as organic acid. So it is stored in the vacuole as malic acid. This is during the night time, and during the daytime, the second fixation of carbon dioxide will occur, which the organic acid, which is the malic acid, will be converted back into malate. Into malate, and then from there malate will split into pyruvate and also carbon dioxide. So the carbon dioxide will enter into the normal Calvin cycle, the normal Calvin cycle to produce sugar. The sugar produced will be G3P. G3P. So from the molecule of G3P, it can synthesize, for example, glucose. So for chem plant, it involves carbon dioxide to fix twice, to be fixed twice, which is the first fixation will occur at night time. The second carbon fixation, which is the Calvin cycle, will occur during daytime. Question 4. In plant cells, ATP synthase is located in the thylakoid membrane and the inner mitochondrial membrane. So the synthesis of ATP requires ATP synthase, which for plant cells, it will be located in the thylakoid membrane and also mitochondrial membrane. So you have to take note also that plant cells also carries out cellular respiration. Okay, cellular respiration. So because plant cell also carries out a cellular respiration to produce their own ATP, so the ATP synthase will also be located in the inner mitochondrial membrane, okay, apart from the glycol membrane, which the plant cells will carry out photosynthesis. Okay, so photosynthesis that during the first phase of photosynthesis, which is the light reaction, involve chemiosmosis, which is to synthesize ATP using ATP synthase. Question number 5. Which of the following does not perform photosynthesis? Obviously, the answer would be green June beetle. Okay, green June beetle is obviously an insect. So insect does not carry out photosynthesis. So fern carries out photosynthesis. Cyanobacteria is a type of bacteria that is capable of undergoing the process of photosynthesis. Same goes to blue-green algae, also carries out photosynthesis. Question 6. During photosynthesis, carbon dioxide is reduced. Okay, so carbon dioxide will be reduced during photosynthesis until it becomes sugar. Okay, sugar. So as you can see, carbon dioxide initially it is CO2, and during the process of photosynthesis, at the end, it will become glucose. Okay, C6H12O6. So as you know from the term reduced, so it means that the compound will receive electrons and also protons. Okay, so this one, the carbon dioxide, will become glucose or sugar. Okay, so for the next question, stomata of chem plant opens at night and closes during the day because loss of water through stomata can be reduced at night. Okay, so the answer will be D. So for chem plants, okay, so these are plants that lives in hot and dry area. Okay, so because the plant lives in hot and dry area, to prevent water loss and to conserve water, the stomata will close, okay, during daytime, and it will open at night time. Okay, so when stomata opens at night time, then the carbon dioxide can enter into the leaves and can be fixed. Okay, during night time, and then stored as organic acid first at night time, and then undergo the second carbon fixation during daytime, which is it undergoes the normal Calvin cycle during daytime. Okay, so basically this is to conserve water. Okay, to conserve water within this chem plant because they live in hot area. Purple sulfur bacteria have a single, okay, it only has one type of photosystem, which is PS1. Okay, it generates ATP during photosynthesis through cyclic photophosphorylation. Because the bacteria only has photosystem 1, so the electron will flow in a cycle. Okay, in a cycle from photosystem 1 through the electron transport chain and back to photosystem 1 to produce ATP molecule. Okay, so the electron will be flow in a cycle. Okay, from P700 and back to P700 of photosystem 1. So question number 9. A net synthesis of 2 molecules of G3P would consume a total of 6 molecules of carbon dioxide. So G3P is the product, okay, formed during Calvin cycle. Okay, Calvin cycle. So G3P is a 3-carbon sugar. So you know the purpose of the Calvin cycle is to produce G3P. So from the G3P molecule, it can synthesize glucose. Okay, so glucose is a 6-carbon sugar. Okay, 6-carbon sugar. So to produce one molecule of glucose, okay, one molecule of glucose, which is a 6-carbon sugar. The process requires the production, okay, the net production of 2 molecule of G3P during Calvin cycle. So in order to produce the 2 molecule of G3P, the process will requires 6 molecule of carbon dioxide. Okay, so 6 molecule of carbon dioxide to produce 2 molecule of G3P. Each G3P will have 3 carbon. Okay, 3 carbon. So you have 2 molecule of G3P. So as you can see, so this is one molecule and this is another molecule. So altogether, that's why the process requires 6 molecule of carbon dioxide. Okay, 6 molecule of carbon dioxide, and from there, the 2 molecule of G3P can be used, okay, to synthesize 1 molecule of glucose. Question 10. Radioactive carbon dioxide is added, okay, radioactive carbon dioxide is added to a suspension of photosynthesizing seaweed. Which of the following will be the first, okay, the first compound labeled with the radioactive carbon dioxide? So the answer will be 3-phosphoglycerate, or also known as PGA. Okay, so it means that basically for this experiment, it is to trace the flow of the carbon during the Calvin cycle. Okay, so basically the carbon dioxide, in this case the radioactive carbon dioxide, will enter into the Calvin cycle, and when it enters into the Calvin cycle, it will be incorporated with the carbon dioxide receptor, which is RUBP. Okay, so RUBP is a 5-carbon sugar. Carbon dioxide is a one carbon compound, and this will produce a molecule, okay, which is a 6-carbon intermediate. Okay, so this 6-carbon intermediate is a very unstable molecule. Okay, so because it is a very unstable molecule, it will break into 2 molecule of PGA. Okay, 2 molecule of PGA, or also known as 3-phosphoglycerate. So for Calvin cycle, so this is the first products of carbon fixation that will be produced. So it is a 3-carbon sugar basically. Okay, so that's why the Calvin cycle is also known as the C3 pathway. Okay, the C3 pathway, because it produces the first product is a 3-carbon sugar. Okay, the first product of carbon fixation is a 3-carbon sugar, which is 3-phosphoglycerate, or PGA. Question 11. Which of the following is not an accessory pigment? The answer would be chlorophyll A. So chlorophyll A is the main photosynthetic pigment of plant. It is the most abundant, and they are found, okay, in the reaction center of PS1 and also PS2. Okay, PS2. So they are found in the reaction center. Okay, so this is what we call it as a special kind of chlorophyll A. But chlorophyll A can also be found, okay, in the antenna, okay, in the antenna complex, in the antenna complexes of this photosystem. Okay, so for example, so this is a photosystem. For example, photosystem 1, or it can be photosystem 2. At the center here, we have a reaction center that has a special pair of chlorophyll A. Okay, and the rest of this region is also known as antenna complexes. So these antenna complexes will consist of many accessory pigments such as carotenoids, xanthophyll, chlorophyll A, and you can also find chlorophyll B, and you can also find chlorophyll A at the antenna complexes. Okay, so basically the photosystem will have 2 major regions. One is the antenna complexes, the other one is the reaction center. Okay. So figure 12, sorry, question 12, figure 1, okay, is a leaf anatomy of a C4 plant. Identify the region where oxaloacetate is produced. So this is basically the leaf anatomy of C4 plant. So the leaf anatomy of C4 plant will have a few types of cell, which is here. A is mesophyll cell. Okay, mesophyll cell. B is bundle sheath cell. Okay, C is vein, and D is epidermis, lower epidermis lah in this case. Okay. Okay, so if you look at the question, it asks you to identify the region where oxaloacetate is produced. Oxaloacetate is a 4-carbon compound which is produced, first from the fixation of carbon dioxide with carbon dioxide receptor, which is PEP. PEP is a 3-carbon sugar. Okay, carbon is a 1-carbon compound, so it produces a 4-carbon compound which is oxaloacetate. So that's why for this plant, it is also known as C4 pathway, because the first product from the fixation of carbon dioxide, is a 4-carbon compound which is oxaloacetate. So this will occur in the mesophyll cell. Okay, so mesophyll cell is A. Okay, so this is where the first fixation of carbon dioxide will occur. Okay, so again, C4 plant will undergo carbon fixation twice. Okay, the first carbon fixation will occur in the mesophyll cell, and the second carbon fixation will occur in the bundle sheath cell. So basically, with the bundle sheath cell, the carbon cycle will take place, which is the second carbon fixation. Okay, so you have to take note for C4 plant. It involves two different types of carbon dioxide receptor, which is PEP during the first carbon fixation, and RUBP during the second carbon fixation, which is during the Calvin cycle. And for this plant, it also requires two types of enzyme to fix the carbon, which is during the first carbon fixation, it requires PEP carboxylase, and during Calvin cycle, it requires Rubisco to fix the second carbon, to make sure that the second carbon fixation occurs, okay, during the Calvin cycle. Question 13. In photosynthesis, the products of light-dependent reaction are, okay, ATP, NADPH, and oxygen. So the answer will be C. Okay, light-dependent reaction is the first phase of photosynthesis, and it involves either the cyclic photophosphorylation, or non-cyclic photophosphorylation. For non-cyclic photophosphorylation, it involves the production of ATP and NADPH and oxygen. So oxygen, as you know, it is the byproduct of photosynthesis that takes place during the light-dependent reaction. The cyclic photophosphorylation will only produce ATP molecule. Okay, so the production of oxygen is basically from the photolysis, from the photolysis of water. So photolysis of water will produce hydrogen ion or proton, electrons, and also oxygen atoms. Okay, half of oxygen atom during photolysis of water. So basically, photolysis of water is the source where the oxygen molecule will be released during photosynthesis. So number 14, which statement is not correct about C4 plant? Okay, so the statement asks you the incorrect statement about C4 plant. If we are going to, okay, so now basically we we are going to look at each statement. Okay, so for statement A, they have PEP carboxylase and RUBP carboxylase. So for C4 plant, okay, so they will use both of these enzymes. Okay, so PEP carboxylase will be used during the first carbon fixation that takes place in the mesophyll cell, and the RUBP carboxylase or Rubisco, okay, will be used during the second carbon fixation, which is the Kevin cycle, that will occur during bundle shift cell. So statement A and statement C is correct. Okay, so for C4 plant, it involves both of these cells. So mesophyll cell is where first carbon fixation occur. Bundle shift cell is where the second carbon fixation occur, or the Calvin cycle will occur. So basically for C4 plant, they have to undergo the Calvin cycle, because the Calvin cycle is the purpose of Calvin cycle is to produce the sugar. Okay, which is G3P, and from G3P it is to synthesize glucose, for example. For statement B, carbon fixation occurs twice, so of course it occurs twice. Okay, first in the mesophyll cell, second in the bundle sheath cell. So statement B is correct. For statement D, carbon fixation occurs in the same cell but at different times. So obviously this statement is incorrect. So the answer D is the answer will be D. Okay, the same cell is obviously not lah. Okay, because as you can see from statement C, it involves both of these cells. Okay. So for question number 15, what is the role of photosynthetic pigments in chloroplast? So the answer will be to absorb, okay, different wavelength of light. Okay, so that is the function of photosynthetic pigments. So these photosynthetic pigments can be chlorophyll A, chlorophyll B, xanthophyll, carotenoids, okay, which are located in the photosystem to absorb light energy. Okay, wavelength of different wavelength of light energy. Okay. So question 16, the electron donor in non-cyclic photophosphorylation will be water. Okay, so water is the source of electrons in the non-cyclic photophosphorylation. So water will undergo the process of photolysis. Okay, photolysis to break the water molecule into hydrogen ion. Okay, and then into electrons and also oxygen atom. Okay, oxygen atom, in this case half oxygen molecule. So the hydrogen ion will contribute, okay, to the high concentration of hydrogen ion in the thylakoid space. Okay, so this is crucial for the process of chemiosmosis. And then the electrons, okay, will uh, will um, will be received by P680 plus, okay, in the oxidized form. Okay, so the P680 plus will receive the electron and become the reduced form, which is P680. So this chlorophyll A in the reduced form will absorb light energy, and the electron of P680 will be energized and released from the P680 and will be accepted by the primary electron acceptor of the electron transport chain before it is passed from one carrier to another carrier to release energy to generate ATP molecule through the process of chemiosmosis. Okay, so oxygen molecule, okay, from this half oxygen molecule, it combines with another half oxygen molecule to produce one oxygen molecule, which is the byproduct of photosynthesis basically. So oxygen molecule is produced basically during non-cyclic photophosphorylation. Question 17. The rate of photosynthesis for C3 plants, okay, decreases when the concentration of oxygen increase. Okay, so if for C3 plant, basically it requires only one type of enzyme to fix carbon dioxide during Calvin cycle. So this enzyme is Rubisco. Okay, Rubisco is also known as RUBP. Okay, RUBP carboxylase. For this enzyme, Rubisco or RUBP carboxylase, it can bind to both, okay, carbon dioxide and oxygen. Okay, and oxygen. If the concentration of oxygen is high, okay, in the leaf, the enzyme will prefer to bind to oxygen compared to binding to carbon dioxide. Okay, so when does this occur? It is occur when the stomata of the C3 plant close. Okay, when the stomata of the C3 plant close, it limits carbon dioxide to enter into the leaf of the plant and to enter into the Calvin cycle to carry out the Calvin cycle to produce sugar. Okay, so when does the stomata closes? It is when the temperature of the surrounding for that plant is hot. Okay, hot. So this is okay. So this is to conserve water within the plant during hot weather. Okay, so but the side effects is that when the stomata closes, the concentration of oxygen will increase in the leaf because oxygen is the byproduct of light dependent reaction. Okay, so when oxygen concentration in the leaf increases, Rubisco will bind to oxygen instead of binding to carbon dioxide, and so this can lead to photorespiration that reduces the yield of Calvin cycle. It reduces the amount of sugar produced during Calvin cycle. Okay, so this is the negative side. Okay, the kelemahan. Okay, kelemahan Rubisco ni, dia akan bind kepada oksigen bila oksigen concentration dalam leaf tu tinggi, okay, berbanding dengan carbon dioxide. Okay, so for question 18, how many NADPH are needed to synthesize one molecule of maltose in the Calvin cycle? Okay, so maltose, as you know, is it is a disaccharide that consists of two molecules of glucose. Okay, so one molecule of glucose is a 6-carbon sugar. Okay, one molecule is a 6-carbon sugar. So two molecule of glucose will involve in the maltose a molecule 12. Okay, 12 molecule of carbon to be present in the maltose sugar. Okay, so therefore the amount of NADPH molecule are required is 24. Okay, 24 molecules of NADPH are required. Okay, so basically you have to understand for Calvin cycle. Okay, for Calvin cycle, okay, to produce one molecule of glucose, it requires 12 molecule of NADPH. Okay, it requires 12 molecule of NADPH. So please refer back to your notes in the slide of 56. Okay, slide 56. You look at your note, which is slide 56. It shows you that the cycle to produce one molecule of glucose, it requires 12 molecule of NADPH. So in this case, to produce one molecule of maltose, which consists of two molecule of glucose, so you have to times 12. You have to times 12 by 2, and then you get 24. Okay, 24. Okay, so question number 19. The reactant of light-dependent reaction, okay, light-dependent reactions are water, ADP, and also NADP+. Okay, so these are the reactants, the molecules that are required during light-dependent reaction to produce the product. So the product from the water, later it will produce oxygen molecule. Okay, ADP will later produce ATP molecule, and NADP+ will later produce NADPH. Okay, so for question number 20, okay, which is the correct sequence for electron flow in the in photosynthesis? So the answer will be A. Okay, so water is the source of electron produced during photolysis of water, and then the electron will be accepted by P680, which is a special pair of chlorophyll A at photosystem 2, and then will be passed along the electron transport chain, which consists of cytochrome complex. Okay, cytochrome complex P700, which is at photosystem 1, and then lastly received by NADPH molecule. Okay, so basically during the flow of electron, the product produced are oxygen molecule, ATP, and also NADPH. Okay, so NADPH and also ATP produced during the first stage of photosynthesis will be required during Calvin cycle to produce sugar. Okay, so question 21. Okay, so which of the following is not associated with the thylakoid membrane? Okay, so ATP synthase obviously are found embedded in the thylakoid membrane. The Calvin cycle is the process that occurs in the stroma of chloroplast, so the answer would be B. Photosystem 1 and also photosystem 2 are also embedded in the thylakoid membrane. Same goes to the electron carrier of electron transport chain, okay, will also be embedded in the thylakoid membrane, so the answer would be B. Question number 22. What is the advantage of C4 plant in sugar cane? Okay, it is to prevent oxygenation of RUBP. Okay, so that is the advantage of C4 plant because it has two types of carbon dioxide receptor, which is RUBP and also PEP. Okay, it uses two different types of enzyme, which is PEP carboxylase and also RUBP carboxylase or Rubisco. Okay, so this is to prevent photorespiration from occurring. Okay, where the Rubisco will bind to oxygen instead of carbon dioxide. So that's what the answer will be B, to prevent oxygenation of RUBP. So that's the end of part A for chapter 6, photosynthesis.